very long way to I'm happy to do that. All right, let's go ahead and get started. So my name's Scott Ransom. I'm an astronomer at the National Radio Astronomy Observatory, and I'm going to be chairing this session. There are 3 talks in this session, and we're going to, the first talk will be 35 minutes, and each of the next 2 talks are 25 minutes, and that's total durations. So for the speakers, beware. So if you don't want questions, you can use all of your time. I'm going to be quite draconian, unlike Jay, because we are way behind. Once again, 35 minutes for the first talk, 25 each, and I will give the speakers 10 and 5-minute warnings. And hopefully we have some time. The speakers should tell me if they want some time for questions. The first talk, we have Lynn Rothschild, who's here from the NASA Ames, and who will be talking about evolution and astrobiology. Lynn?
Thank you very much. Many of you who know me probably think I come from the outer solar system. In fact, I come from Connecticut, so if there's one thing I can do, it's talk fast. So I'm going to go full Connecticut Yankee on you here today. So I was asked to actually originally talk about evolution and astrobiology, so I wanted to start with a few thoughts about that, because I know evolution means different things to different fields. But for biology, ecology is sort of a snapshot in time of what happens, the interaction between organisms and their environment. What evolution does is it adds the time component. So it's like going from a still picture to a movie. If you don't laugh, it means you can't read my slides. This is a test. And Dobzhansky very famously said that nothing in biology makes sense except in the light of evolution. He said that in an essay in 1973, and I'd like to make the point today that nothing in biology actually makes sense except in the light of astrobiology, because we're taking not just the time component of evolution, but also the spatial component and asking questions, could it happen again? Did it happen elsewhere? And how could that possibly be? So for evolution to be truly useful to astrobiology, it must be predictable. It's not ever going to be quite like the laws of gravity and so on, but there's got to be some predictability to it, because if you just say, well, this is what life's like on planet Earth, I have no idea what it would be like anywhere else, how can you go search for it? You have to have at least some hypotheses. But the problem is, of course, we only have one actual narrative, one data point. And I always like to say it would be sort of like if I went into the chair of an English department and said I would like a faculty position as a tenured professor in English, and they said, all right, which books are you familiar with? Well, I read one once. They would laugh me out of the room. But that's the situation we're in with biology. We have one book and we have one narrative. So we have to sort of be a little sneaky about this question, is evolution predictable? And of course, you have to start with questions like, what do you, thank you very much. And I'm often accused by, the legal people that I need to cite it when I use stock photos. This is not a stock photo, this is my parents' last dog, Pippi. It's no longer with us, but she was adorable. Anyway, so the question is, what is life? Because if you're going to look for it, you've got to start with what is life. And I really love Carol Cleland's point, I'm borrowing this shamelessly from her, that Da Vinci, when asked to define water, for example, said water is sometimes sharp, sometimes strong, sometimes acid, sometimes bitter, sometimes sweet, sometimes thick or thin, sometimes it's seen bringing hurt or pestilence or hepatitis B, sometimes health-giving, obviously I'd lived there, and so on and so forth. If anyone in this room were asked to define water, it would be very simple. And so to me, the key question is there an H2O definition for life? Because clearly at this point we're at a Da Vinci level trying to describe life. Now you have to realize that maybe it's because the science hasn't gotten there, but it may be that water is not life is not a natural kind like water, and we will never have an H2O definition, which as an aside means that if it's a matter of taste, an alien might not define us as living, and there could be consequences of that. So we need to at least have some kind of operational definition. And I think one of the key things to remember here is that life is a process. It is actually not a thing. It's sort of like the difference between a pond and a river. A river, by definition, is no longer a river if the water isn't moving. And the same thing with life. If these processes aren't going on, you have a corpus, but you don't have life. And so we have a process, and the process includes fighting entropy, a la Schrodinger. I just was at a meeting with Steve Benner, 300 people in the room, and we were the only two who had actually read Schrodinger. That was horrific. Fortunately, I know I'm among fellow people who have here, so we don't need to explain it. And then I think this is actually a very key point that I firmly believe, and there may be disagreement in this room, but I firmly believe that organic carbon is basically non-negotiable. And then you need some kind of solvent like water. So why am I so keen on carbon? Well, it's the fourth most common element in the universe, so there's lots and lots of it. This is what they call the astronomer's periodic table. I only wish that biologists had been given this option, but it does show you right away that there's vastly more hydrogen in the universe than anything else. Helium is sort of a non-starter for biology, with the exception of party balloons or something. And then you've got oxygen, carbon, nitrogen, and so on. And so you can see that you're immediately actually talking about elements that are very commonly used by biology. So there is sort of some mapping between the ones that are pretty common and the ones that biology uses primarily. And of course, biology doesn't just use carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. That we are actually metallo-organic compounds. Over 50% of our proteins actually have a metal cofactor. And people ask me, well, what metals are you talking about? And the easy answer is look at the back of a vitamin bottle. So I've conveniently provided the back of a vitamin bottle for you. And you can see here the scientific version of the back of a vitamin bottle. There are lots and lots of these. And so you can start to get an idea of what the building blocks might be here and potentially, therefore, elsewhere. Then the last thing, which I find absolutely stunning, is our colleagues at NASA Ames and Leiden and around the world have found, of course, these carbon molecules out in the interstellar space. And what is stunning is that these include the building blocks for life here, things like amino acids. And so I am always sort of had chills down my spine when I realize that if there's a student around Alpha Centauri having to go through the same kind of pre-med weeder biochem class that I did, they also may be using sort of the Alpha Centauri version of Lehninger or Stryer or whatever people use today. So we have the building blocks that not only are used for life on Earth, but it seems to be somewhat of a common language out there. And, besides, if you want to find a place not to use a carbon-based life, we've got it right here, third rock from the sun. Here's planet Earth, and here's a very simplistic pie chart I made from the source of all knowledge, the Handbook of Chemistry and Physics. And you can see that the vast majority of the crust is made of silicates. There's very, very little carbon percentage-wise in the Earth's crust. And so if you wanted to pick a planet that would be a great place to have a silicon-based life form, here we go. And as we move on in age and time, I'm sure we will become silicon-based life forms. But at least we have the conceit right now that we still are dominating the silicon-based life forms. So if you follow me so far, there are all sorts of things that we can derive from these first principles, because organic carbon has certain properties. All organic carbon, for example, absorbs in the ultraviolet range, and therefore, if every organism is based on organic carbon, you're going to have a problem with ultraviolet radiation, and that may say something about your relationship to your star. So, for example, this is the narrow part of the electromagnetic spectrum that life uses. So we don't want to go too much into the UV because it is extremely dangerous to our biomolecules. DNA has peak absorption at about 260, proteins at about 280 nanometers, so you get anywhere near there, you've got problems, and of course there are lipids and other things in your cells as well. On the other end, as you get into the infrared, there's not a whole lot of energy. So yes, there are some organisms like pythons that may see in the infrared if they're trying to follow a mouse and to eat it at night. But in general, that part of the spectrum is avoided because there's just not enough energy. And so you end up with plants, for example, using that 400 to 750 nanometer range for photosynthesis, because that's where the action is without being too dangerous in that UV. And it's that same range that we use for vision, we animals on planet Earth. So I would argue, just based on what we know about organic carbon chemistry, that this would be the window that you might want to look anywhere. So you can see what I'm trying to do is aim for, agnostic ways to look at life just based on something, for example, organic carbon. And we could, go down this route further. But let's switch over to the solvent for a moment. And of course, we use water. A couple of years ago, I was the opening speaker for a water congress in the Netherlands, and I had 500 reasons that water would be good. And I'm going to boil that down to the fact that it's a terrific solvent because, unlike most other obvious solvents, it's liquid at a fairly warm temperature, or 0 to 100 at our atmospheric pressure. It's also got great chemical properties for reactivity of organic carbon molecules. It's unusual in that it floats when it's solid rather than sinks, and this gives you the ability to have these ice-covered moons like Enceladus and Europa. It also gives you the ability to have a gin and tonic with a nice floating ice cube. So there are all sorts of great things with water, and there's, of course, lots of it. We go back to the astronomer's periodic table. Oxygen's number 3 in the universe's catalog of top, 10 elements, and hydrogen's number 1. Here's an artist's conception of actually the origin of another solar system, but it probably could apply to ours as well. Tons and tons of water vapor. Of course, Titan was put into our solar system to test astrobiologists' faith in liquid water. You can see I've been spending too much time at the Vatican Observatory. And certainly there have been attempts to look at the potential for an organic carbon solvent as an alternate possibility. I think in general, people are shying away from it and sort of focusing on the fact that Titan does have solid water on the surface. You can see some of it there, and potential for liquid water beneath the surface. To me, I think one of the real problems with a life form that uses something like ethylene or methane as a solvent is that it is so cold, and physical damage, for example, from ultraviolet radiation, ionizing radiation, is still going to occur at the same speed, but your ability to repair the damage is going to be so dramatically slower that it's very difficult for me to conceive of how a life form can actually make it at those incredibly low temperatures. So let's get back to life. We also have a problem with the selective tyranny of the environment, and I've been alluding to that you don't want to go too much into the UV because of the danger, and you don't want to go too much in the infrared because of the lack of energy and so on. There are other sort of environmental factors, for example, temperature, pressure, desiccation. If you've got water as a solvent, then desiccation is an extreme environment. Radiation, been alluding to chemical extremes, and certainly oxygen. I realize that most people don't put oxygen on their list of extremophilic environmental conditions. I certainly do, because we are acutely adapted to deal with high levels of oxygen, about 21%-ish in this room. But while oxygen doesn't really hurt us, and the byproduct water doesn't, it's those reactive oxygen species in the middle that are so incredibly dangerous and can cause oxidative damage, and they've been implicated in everything from diabetes to on and on and on. And so this is really a problem being an aerobe. And at this point in the talk, I usually say, do not stop breathing, because we wouldn't be here if we weren't aerobes. We're big, energetic creatures that need the aerobic metabolism. But it's actually a very dangerous game. And if we were a conference of anaerobic bacteria, we would be looking at ourselves and saying, what are they thinking? They are living in a ridiculous environment. And so I think we shouldn't be too anthropomorphic. We actually are in an unusual environment. And the implication for this is that the astronomers and planetary scientists in the room who are working very hard to find us biologists a planet that has free oxygen, don't bother. Really. I mean, it would be great for us, but in terms of life, it's actually much more difficult than if you're in an anaerobic planet. So We could also look at potential for natural selection, which gets sort of contorted into various NASA definitions. And just to remind you, natural selection, since it's the off season from Darwin's birthday, is that you're going to look similar to your parents because we have heredity, and that not all of the offspring will survive, and therefore you've got a changing shift in the statistics of each ensuing population. And that's all it really is, and it's a great principle, and I would be very surprised if it didn't happen elsewhere. I also realize that there is no ridiculous mechanism for evolution you could possibly think up that there is probably not some insect in the Amazon that does it. There are lots and lots of other ways to do it. It was not natural selection that had the dinosaurs go extinct. It was just the ultimate bad hair day. So there are other ways for evolution to occur, neutral drift and so on. So we've talked about organic carbon, we've talked about solvents, we've talked about, well, universality of chemistry and physics laws. I realize that funky things happen at the edge of the universe and so on. But, you know, in general, it's not going to be grossly different elsewhere. Likelihood of the availability of inputs, and we'll talk more about that in a moment. And if you take all these sorts of things together, you can have some kind of near-term predictive power, especially once you look at the constraints of ancestry, genetics, and developmental biology. I would be shocked if any of you told me that your children had wings, because you don't tend to make those sorts of leaps. But, you know, there are certain constraints, canalization in biology, that you can have an awful lot of predictive power. So let's try just a couple of examples. For example, if you look at the amino acids that are used for life, and then you rank them as Higgs and Pedretz did into their concentration meteorites, stardust analogs, prebiotic experiments, and so on, you get the same sort of ranking. And so that suggests that glycine, which is vastly more abundant than everything else, is probably going to have a key role in life. And then you start to go down from there. What about peptides? It turns out that you can even make peptides by shock synthesis. So these are mechanisms that should be very widely available in our solar system and beyond. The ingredients for life, the basic polymers, lipids can certainly form abiotically and self-assemble. As I mentioned, amino acids are certainly formed abiotically, and even short peptides. Certain parts of RNA bases can be found abiotically. You're not getting actually ribonucleotides abiotically yet, but then if you do actually make those, you can polymerize them abiotically without any trouble. DNA is not quite so easy, which is why I feel like if we find a DNA-based organism somewhere else, that's almost proof positive that our instruments were contaminated, but that's another discussion. So to end sort of the first part of the talk, if you start to look at some of these questions, and if you buy the argument that at least you're constrained by having life forms based on organic carbon chemistry and using water as a solvent, then there are all sorts of things that would fall out from that in a very general sense. So that certainly then constrains your search space for life elsewhere, and what would have occurred if you, we used to say replayed the tape, but when you have younger audiences, they don't know what tape means anymore. I trust everyone in the room here does that. So that's one approach, and so the last part of the talk, we're going to flip to another approach, and that is once you have life, arguing from something that we call in biology convergence. So everyone, I'm sure, recognizes this as a picture of a cactus. For those people at the Santa Fe meeting a few months ago, did that. And here's another organism that uses thorns. This is, of course, a rose. And this is a little trickier. This is a whistling thorn acacia from Africa. And that one, I know some of you have seen this before, but yeah, it's the rear end of a porcupine. But all of them have converged on exactly the same way to say, don't think about eating me. It's going to hurt. So that's convergence. This is not direct descent, this is convergence, coming up with the same solution to the same problem independently. And I would argue that many of these, I like to call great moments in evolution, also are conceivably convergent, which means that there's at least a possibility that if we replay the tape, some of these things would happen again on the Earth and elsewhere. So for example, I gave a talk a few years ago at the Royal Society with the question, could photosynthesis evolve again? Well, photosynthesis is just autotrophy, taking inorganic carbon and turning it into organic carbon using light as energy. Our sun is virtually limitless in its source of power. I'm sorry, aside, but I have no patience for people who say, well, Rubisco is inefficient and photosynthesis is inefficient. You know what? There are lots of photons to go around. It's okay. And then if you combine the two, you get photosynthesis. So what do you need as preconditions for photosynthesis? Well, you need energy in the form of light, you need inorganic carbon, and then you've got certain biological precursors. And there's certainly plenty of photons available here, and there would be available to many of the places that we're talking about that are revolving around a star. In fact, every place I think we're talking about life originating has a parent star and has the ability to at least somehow pick up some photons. But what about the biological precursors? Well, this is where we look at this question of convergence. Every one of these molecules can harvest light. And it doesn't take a trained chemist to see that their structure is dramatically different. They've all converged on the ability to harvest light. So light harvesting compounds arose multiple times. Reductants multiple reductants are used. Oxygenic photosynthetic organisms like algae, cyanobacteria, and plants happen to all use H2O. They use water, but there are lots of bacteria that use many other reductants. So again, it looks like this is sort of convergent, that you're not really stuck with one. And this is actually what my postdoc proposal to come to Ames years ago was on. How much of the carboxylating enzymes are convergent, or are they monophyletic? And we haven't done a really good study using the molecular data. I hope to do some more of that this year. But I will tell you that a few of them use the same trick of having a cofactor. But many of these, I would be willing to bet anything, are convergent. The ability to take CO2 and add it to another molecule. Because in here you see everything from photosynthesis to fat synthesis. These are clearly different things that are not even found in the same organism. So the quick way to sort of summarize that is, yes, if the ingredients for photosynthesis would be available elsewhere, and the evolutionary pressure to be able to make your own organic carbon, if you're organic carbon based, would be there, and that at least on Earth, all the prerequisites from the biological side are highly convergent. So you can play the same sort of game with things like the origin of the eukaryotic cell by primary and secondary symbiosis. And this has happened multiple times. There are multiple groups of my friends, the protists, who have acquired chloroplasts at different times, different ancestries, and so on, reduction in different ways. So you've got the cryptomonads and the euglenoids and so on. So they've all done the same sort of thing over and over. It's just that one set ended up going into the chloroplasts in plants. Same sort of argument with things like multicellularity. Here is an overall tree of life from Norm Pace's lab. It's quite old, but it still gives you the idea that the vast diversity of life, at least if you look at ribosomal RNA, indicates that multicellular taxa are a very, very tiny part of it. So they're the fungi, the plants, and the animals. It's just that little orange tip that most organisms are unicellular. Why would something even want to become multicellular? Well, if you're larger, it's easier to avoid prey if your prey is still microbial. You can get away from diffusion problems of gases. This is something that would occur any planet or moon you go to. You're still going to have diffusion problems. So if you have lots of little cells, you can get the gas into your cell, whereas if you have one giant cell, you've got a huge problem interacting with your environment. You can then start to coordinate. So maybe you could start to move like a volvox or a fish. You can have senescence, you can have individual cells die and your whole organism doesn't die. You can sequester your germline. I hope you're feeling pretty good about being a multicellular organism right now. So there are all sorts of great parts about being multicellular, so it will not surprise you to know that that's convergent. And in fact, I'm a little unfair because a lot of these protistan taxa, I believe, are functionally multicellular as well. Transition to land, that's very likely to be convergent. We think that the major transitions with plants and animals occurred in high altitude lakes. It had to do with the solubility of gases in water, but there are many other organisms that have at least tried to make this transition to land independently. It seems to be highly, highly convergent. Human skin color is not a genetic badge. It has to do, again, with interacting with the environment. It is highly convergent. Here's a map of solar radiation on planet Earth, and that pretty well maps human skin color, you know, allowing for the fact that high altitude also really encourages a lot of melanin production. Again, I can go into a lot of that, but a lot of that was, it's been done by Nina Jablonski, used to be at the Cal Academy, has been Penn State for many years. Fascinating story that she's come up with there. Intelligence, sorry, but I also think that that's highly convergent. And I won't take a lot of time reminding you of current mammalian phylogeny, but the Eutheria divided off into the Atlantogenata and various other taxa. You've got the Afrotheria and the Xenarthra that split 103 million years ago. They're the Afrotheria in Africa and Xenarthra. And then you've got the Boreoeutheria coming to the New World, and the Lauriaceathera, and so on and so forth. And really the point of showing you all this is not to watch me try to pronounce long words, but to show you a rough idea of how mammalian phylogeny goes. And so if you start to look at intelligence, what's interesting is you see that it is highly convergent. And in fact, you could make an argument that there are other very intelligent creatures on planet Earth that drive home the point of convergence and intelligence. And you can play the same game with aquatic mammals. That is also highly convergent. Yes, I am jealous that they have manatees at Kennedy Space Center and we don't at Ames. We only have turkeys and hares, but nonetheless, they're the aquatic mammals. Fossorial mammals, those of you who know me well, pocket gopher is on my top enemy list right now, despite the cute picture, but pocket gophers, moles, golden moles are all fossorial. They go under, dig underground. You've got ant-eating mammals that are highly convergent. So finally, I'm going to ask the question, can we now move forward beyond what we've been given to look at and use something like synthetic biology, which I'd like to say is the art of the possible in terms of astrobiology, and start to look at alternate possibilities. So synthetic biology is just the design and construction of new biological functions and systems not found in nature. This is probably a reasonable definition, but we are getting to the point that we could also start to look at forming life de novo. And therefore you can ask, can we make a 6-letter alphabet? Do we need to use DNA? What about XNAs, which it's great xenonucleic acids, which various people like Vitor Pinheiro and now in Belgium's working on. So that was sort of a lot to just give you a flavor of the way you could approach this question of is evolution predictable? And my conclusion is if you buy the carbon and you buy the convergence arguments and water as a solvent, yes, we would still be organic chemists because of these arguments. And so that at least constrains our search space, not perfectly down to little green men that are about 3.5 feet high, but by golly, that did constrain us quite a bit and gives us a good way forward. Thank you very much.
We do definitely have time for questions. Yes.
Well, who's been waving their hands at me the whole time?
Yeah. So do you want to say anything else?
Sure. No, that's okay.
Go.
I can go back and do it slower though if you like.
I'm Ted Peters from Berkeley. Thank you for a marvelous defense of convergence theory. My question is, if you're going to move from the defense of convergence theory to the expectation that we'll meet aliens on exoplanets a lot like us, do you need to add anything? And I raise this because Simon Conway Morris has a convergence theory a lot like yours, but he doesn't expect to meet aliens like us, and so do you find him self-contradictory, or is there something you need to do to add to convergence theory to say, we know what to expect on an exoplanet?
So first, I have to actually, to be perfectly honest, the whole reason I got into this business, I did this talk, is because Simon Conway Morris invited me to a meeting years ago at Windsor and asked me to do this.
Oh, good.
So he and I are actually very similar lines. And then you can start to play a, I hate to say this, looking at Frank, a Drake equation sort of game and look at the probability. I've just told you what's possible. And that's where, the convergence comes in. But I believe that the synthetic biology is what allows us to start to get a handle on how probable something might be. Before I got into synthetic biology, which is the other half of my life now, I really had no idea what a philosophical difference there was between science and engineering. And science is looking at what we have here, and that's astrobiology in general, but now I realize when you go into engineering, it's kind of that. Now I can look at all the things that have not been made and see why we don't see them today. Maybe it's just because, and maybe there's not actually, there's a good reason. So Simon and I actually are very much on the same line, but the number of steps to come up with me standing here all the way through, as I said, DNA, I think was a kind of unlikely step, and so on. I wouldn't even want to put probabilities on it, but I suspect mine's a little bit lower than Simon's.
Thank you.
All right, in the front here, and then we'll go in the back.
So, I've sometimes heard it said that it's possible unicellular life, or simple, quote, simple.
Oh, no, don't ever say simple. I'll tell you a story of drinks tonight. Don't ever say simple. There is no such thing as simple life.
Quite common in the universe, but there are reasons to think that multicellular life might be rare, and they'll point to things like the late date of the Cambrian explosion. And I'm trying to square that with your statement that multicellularity seems to be
see, I don't believe that. I don't believe. See, to me, the enormous gap is between non-living and living. And once you've got a life form, you're off to the races. And multicellularity did occur over and over. Whether we can see it in the fossil record or not is a totally different situation. And people who, for example, work on the origin of eukaryotes, when you look for a eukaryotic fossil, they set the stringency so high so that there wouldn't be any ambiguity. But in fact, there's a huge overlap in size between eukaryotes and prokaryotes. But anything that's an overlap, they won't count because it's ambiguous. So I actually believe because that gap, to me, is so much smaller than between living and non-living, I am willing to bet eukaryotes arose really early, and I'm not the only one, really, really early. And we just don't see them in the fossil record.
In the back, and then...
So I agree with your arguments and your conclusions, but there's something that keeps bugging me in the back of my mind.
We're out of time.
Your conclusions is what I would call anthropocentric, and anthropocentric hypotheses have a poor historical track record.
In what respect?
It's saying that...
in what respect do you find my argument anthropocentric?
Not your arguments, but the conclusion. Saying, for example, that life elsewhere is like us.
I didn't say that. I just said based on organic carbon. And that's therefore there are certain things that you can predict if you're based on organic carbon. And that's based on physics. That's based on vulnerability to UV and everything else. That's not based on the fact that I'm a human.
Right. So the example that let me draw an analogy here. We used to think that extrasolar systems, planetary systems, are in some sense similar to ours, in particular that hot Jupiters are impossible, and yet nature surprised us. So that's another example of what I would call an anthropocentric hypothesis. So why is there a good reason to think that nature would not surprise us here?
In fact, I was going to make that very point, but when I was told it was 35 minutes, I took it off. So this is what it was like when I first got to Ames. One solar system, one life form. This is what it's like now. So I'm acutely aware that we are stuck, that it could be grossly different. My point is that if it is organic carbon, I think there are good reasons for that. I think Lee Cronin would like to, sort of push on some high pressure, other things, and I'm not going to argue chemistry with Lee Cronin ever. But if you buy the organic carbon, again, I went through why I think that that's true, there are certain physical things that are going to come out of it. There's certain wavelengths we're going to use, and therefore certain places you're going to avoid, and certain temperature constraints and so on. So, like I say, I'm a little sensitive to that question because I'm trying to be as non-anthropocentric as possible and as completely, secular as I can be, and agnostic.
Dan?
I wanted to follow up about Jason's question. You said maybe the timeline's wrong, that multi-cells could happen a lot earlier, but also you hinted at something that I was a little surprised at, that could have happened many times, that independent origin of multicellular creatures. Is
Absolutely.
there evidence for that?
Absolutely. Well, I mean, plants, animal, and fungi are, prima facie evidence. Little known fact, I was once the president of the Society of Protozoologists. I had a great slide that I took out that every group that we consider unicellular, I would consider temporally multicellular because they have up to 27 different stages in their life cycle. So there are very few, at least eukaryotes, that can do everything within the confines of one single cell morphology.
Well, my question is about separate...
let me, so this, again, I wish I hadn't taken the slides out, but this all works fine. So there was a great paper published 2012. It was Will Radcliffe's thesis work with at the University of Minnesota. And what they did, and this was an incredibly stupid experiment that they couldn't believe, I know there's some NSF people here, they couldn't believe NSF would fund, and they did. And it turned out to be stupid in the most marvelous way because it was simple-minded, direct, and it worked brilliantly. What they did is ask themselves how easy it is to become multicellular. So they took yeast, and there's the ancestral yeast. These are their micrographs. This is Baker's yeast, Saccharomyces cerevisiae. And it's unicellular, it buds, and so on. And they shook it up in a test tube, and they sat there with a stopwatch, and the ones that sunk to the bottom first, they took out with a pipette and passaged them into the next tube. And over the course of 60 generations, they got all these heritable multicellular phenotypes. So they called snowflake phenotypes. Now, there's some question that yeast are secondarily reduced, and in fact, their ancestors were multicellular. So they've tried games like this with Chlamydomonas, which may also have the problem, and so on. So, I mean, to me, though, that was astounding that they could take something that is never found multicellular in nature, and in 60 passages, not 60 generations, but 60 passages, got stable, heritable multicellular phenotypes. That's how easy it is.
Did they find 60 different mutations in the gene lines?
No, they're working on, going through a lot of these. It's not, the 60 was only to give you an idea how quick it is, and you're dividing every hour and a half at 30 degrees, and, you wait till you get a certain OD, and then you do your next one. So,
But I still think it's different. Arguing it's quick is different from arguing it happened independently many times.
But this was just, this would be a third example or a fourth example,
Okay.
that we know the animals, plants, and fungi all did it independently.
All right, we need to move on, but if Stephen Dick can come up and start setting up, and while that's happening, you can ask your question.
Yes, based on the same kind of chemical elements that make up our organic chemistry.
Whoops. Yeah, okay, and so he's got one more question for you.
Do you want to, okay.
He's got one question for you.
Sorry, we're talking down.
Good.
So based on the same kind of chemical elements that build organic chemistry that we have on the terrestrial biochemistry, they are theoretically 10 to the 33 to 10 to the 180 possible organic molecules. And this terrestrial biochemistry is only a very small fraction of what is theoretically possible. So a whole different biochemistry can be built using different nuclear bases, different amino acids, which we know exist. So once you branch out, the outcome will be totally different from what we have available.
Certainly, but for example, amino acids, as I pointed out, that you get this diversity, and I actually had a slide that shows what you find in meteorites that includes the non-natural, the ones that we don't use. You still get vastly more glycine than everything else, for example. You can start to have a pecking order, so it's not that it's a sigma catalog that's just mixed in even amounts in a stew. There should be a huge bias to certain ones, and some of that, I believe, is what we now see in our bodies, and that this would happen over and over again for those reasons. I don't want to take up any more.
All right, well, thank you very much, Lynn. And our next speaker is Stephen Dick, the former NASA chief historian.
Okay, thank you very much. It's great to be back here at Green Bank, especially on the anniversary of the Apollo 11 landing, which I think took place tonight. Well, the first footsteps at about 10:56 p.m. I was, as Martine said, I was one of those millions of kids who were influenced by that event. And it's also great to be here at Green Bank because I was also influenced, even down on the farm, by Frank's searches down here, just down the road for as part of Project Ozma. And those two things came together, of course, at NASA eventually, as we heard from Ken, and hopefully will come again together with these idea of technosignatures and some of the events that are going on now. So we've had two great talks about Barry Blumberg from Carl and Martine. I wanted just to emphasize what a great friend Barry was to NASA history and to the humanities aspects of astrobiology. Barry would occasionally drop by my office at NASA headquarters, the history office, and one day he even brought Martine, and we did a lengthy oral history interview, which is in the archives at NASA, along with many, many other oral history interviews if you ever need to use that resource. Barry, as I say, was a great friend of humanities aspects, and that's one of the reasons that the Blumberg Chair in Astrobiology was initiated. We have, here are four of the, we're up to 7 now, here are 4 of us. David is here as the inaugural chair, and you see some of the others there. I can't emphasize enough what a great position this is if you're interested in doing research on the humanistic aspects of astrobiology, very broadly defined. You spend a year at the Library of Congress. This is funded largely by NASA. I show this not because that's the Blumberg chair there, but because this is the Kluge Center for scholars in the Thomas Jefferson Building, which is just a magnificent building with all kinds of things always going on, and somehow you have to, it's almost information overload. There's anything you want is there. If anybody here is interested in doing that kind of research, please do apply. I think the application is usually due by the end of the year. One of the things that the Blumberg chairs do is to have a symposium. In my case, back in 2014, it was preparing for discovery, a rational approach to finding the impact of finding life, whether it's microbial, complex, or intelligent. You'll recognize some of the people here. Barry Wojtek was attending. Lots of people from the social sciences, humanities, but also the sciences. You see Seth Shostak back there, and David is back there. This resulted in a volume published by Cambridge. 22 authors, very broadly based, philosophers and theologians, one of whom, you were not there, I think, Ted, but anyway, we're going to hear about some of the theological impact later in this meeting. So this is a good entree into the subject if you're interested in this the idea of impact. I did my own book, my own personal take on this, which just came out last year. And there again, you can see the 3 parts of the book and the very broad kinds of things that I addressed there, again, published by Cambridge last year. You might ask, why should we do this? Why should we worry about this? Why not wait until it happens? Then we can worry about it. Astrobiology and society issues are very important. The National Academy, all the way back in 1962, realized that already. What is at stake is a chance to gain a new perspective on humanity's place in nature. There are all kinds of questions about, what our place in the universe is and that sort of thing. And it's important to think about these things ahead of time. As the World Economic Forum has stated, the discovery of life beyond Earth is one of the 5 sort of X factors, and we really need to consider ahead of time what we're going to do in case of such a discovery, and the question is how to do that. Also, the NASA astrobiology roadmaps from the beginning have recognized a broad societal interest in its endeavors and the importance of looking at what the societal impact might be. And in fact, this is part of, in general, the impact of science on society, which I think is very important to do. It's been recognized in the Human Genome Project, for example. There's a social sciences aspect in nanotechnology, artificial intelligence. And when I was at NASA headquarters in the history office, we initiated this series in societal impact of spaceflight. It's actually part of the original and still there NASA Space Act of 1958 that we're supposed to be doing this sort of thing, but we really haven't done much of it until the last decade or so. So these books in this series are all free online. If you just Google NASA history, you'll find these books along with many, many other kinds of books having to do with NASA history. So how do we do this? What are the approaches? That was my first question when I arrived at the Library of Congress. I came up with 3 approaches, not necessarily all the approaches, but these are the ones that I concentrated on: history, discovery, and analogy. In other words, there's a history of examining of the reaction when people thought that life had been discovered. The nature of scientific discovery, I think, is very important for this aspect, and analogy, the use and misuses of analogy. I have a whole chapter on that, and I always emphasize that you cannot make predictions, right? We're not in the business of making predictions, but we can have certain guidelines if we think about these things ahead of time. Okay, at least 6 times, probably more, we thought we had found life beyond Earth. Here are 6 of those. I'm going to just talk about a little bit about the first 2, but more about the last one there, the Mars rock, because I think it's the best analogy. The moon hoax slash satire of 1835, then the War of the Worlds broadcast, which everybody has heard about, and the famous Mars rock, which we heard mentioned earlier. Well, this is 1835. This is what actually appeared in the New York Sun. Amazingly, people believed this. This was a series of 6 stories published in the Sun in 1835, claiming that Sir John Herschel had actually observed this on the moon from with his telescope in the southern hemisphere. Well, by this time in the mid-19th century, scientists knew that life couldn't exist on the moon. There was no atmosphere. No telescope could have that kind of resolution. But the general public believed it anyway. So you might say, why did the New York Sun publish this? Well, it was because they became the most read newspaper in the world. So there is that factor about the media. So I think the lesson there is in the short term, at least, facts won't matter. What the media says will matter. The perception will matter, not the reality, at least in the short term. And again, this is usually seen as a moon hoax. It was actually written as a satire, but people believed it. The next one is the famous War of the Worlds broadcast, the Orson Welles broadcast, which is often seen as initiating a great panic, right? Now that scholars have looked at it, they've determined that there really was no panic, at least not any kind of widespread panic, even though things like the American Experience on PBS still talked about a great panic. As I say, the scholars both of media and journalism and other areas have shown that this was an irresistible opportunity for print media to rebuke the upstart rival radio media, right, for inciting the public. So one of the researchers concluded when circumstances are right, the media can create panic and other effects that are unpredictable, disruptive, and wide-ranging. So that's 2 strikes against the media now. And this is, of course, well, let me move on to famous Mars rock here. What are the lessons that we can learn from ALH 84001, which was mentioned before? Many of you remember back in August 1996 when this took place. And people often say to me, if we discover microbes, I don't think that will have much reaction. These were not microbes, these were nanofossils. And let me show you some of the reactions. Some of you will remember this reaction from your own personal time, right? Here's NASA headquarters on the day of that press conference, which was mentioned earlier. It's just, I think, an astonishing snapshot of the excitement at the time. I've been in NASA headquarters auditorium many, many times, but I've never seen anything quite like this. Here's the little piece of a Mars rock, David McKay, Everett Gibson, and some of the other people here, and you can just see the wild media frenzy at this event. Here are some of the authors of the paper that was published, and standing here with Dan Golden. See their names down there. Mars rock fallout. So first of all, there was a rushed press conference. Things were leaking, which is what is going to happen, I would say. So this was a rushed press conference. All the major TV stations broadcasted live. So there's a lesson to be learned here about staging and content and how to do this, and these are things that need to be thought about ahead of time. Another aspect of this press conference was that you had a skeptical scientist there, Bill Schopf, right, who invoked Sagan's famous dictum, extraordinary claims require extraordinary evidence, right? This was certainly an extraordinary claim. The question is, was there extraordinary evidence? The authors of the paper would say, well, there are 4 or 5 independent arguments that lead to this extraordinary claim, and we consider that extraordinary evidence. So the question, the philosophical question here is, if you have 4 or 5 weak arguments, does that add up to one big weak argument, or does that add up to a substantial extraordinary argument? So we can talk about that, but this is the kind of thing that went on for years after this discovery was announced. President Clinton gave a statement on the South Lawn of the White House. The media reaction, CNN, biggest discovery in the history of science. Headlines in all the news media. Theological implications were raised. Does this confirm the expansive nature of God, or the opposite? And we'll hear more about this from Ted Peters later in this meeting. Within 5 weeks, there was a congressional hearing about life on Mars. Within 4 weeks, Vice President Gore convened a space science symposium. And over years and decades, the fallout to research opportunities in terms of new budgets and new research directions, including things like the Origins program, the Astrobiology Institute, some funding from NSF. So there are real implications to these things, and I think we can talk about the analogies and the disanalogies between then and now. And remember, this was before social media. Can you imagine now with social media how this will spread like wildfire? I want to say just a few more words about the symposium. Obviously, there was interest at the highest levels of government with that statement made by President Clinton, but there was also this symposium, which I think Carl mentioned. And here you see, I've, through the Freedom of Information Act, obtained some of these documents from the White House. Here's the Assistant to the President for Science and Technology, John Gibbons at the time, inviting colleagues to this meeting on December 11th. It was quite a small meeting, probably about 20 people around the table. And here you see it. I think this image has not been seen before. It's in the National Archives, but you see the ambiance here. It was close to Christmas, so we're around the Christmas tree here. This is in the old executive office building, just across from the White House where the Vice President has his office. You see Vice President Gore there on the right to his, on this side of him, the Director of OMB, the Science Director, Science Advisor to the President. You see Dan Golden to the left there. That's Amelia Sargent from the National Academy. John McCall, and there's myself for some reason, and Lynn Margulis, Wes Huntress. And on this side, very interestingly, you have people representing theology, social sciences, and other areas. It was not, this was not primarily a meeting about the science. It was more of what is the impact of this. By the way, Stephen Jay Gould was sitting on this side. And Carl Sagan was supposed to have been here, but he was about a week away from dying. So he was not here, but it was quite an event, I would have to say, like an intellectual salon with the Vice President shooting questions around the table at the various people. And I think, this again is an analogy that we might consider about what will happen at the highest levels of government when this discovery is made.
Any recordings?
No, that's a great question. I have not found a recording or a transcript of what was said. Whether it exists, I just haven't found it yet. Yeah. So, just to summarize on the Mars rock, you can see there the reaction of government institutions, media, and public, along with the reaction of scientists who will subject the discovery to withering criticism, as we always do, right? The media will play an important role, especially now in the age of the internet and social media. But as the Mars rock demonstrates, the final conclusion will be far from immediate. They argued about this, right, for years, and some people, the consensus now is that it's not nanofossils, but some of the people who wrote the paper still argue that it is, right? So, and this is where we get into the idea of discovery. Discovery is always an extended process, and here you see how I sort of parse the anatomy of discovery, detection, interpretation, and understanding, those kinds of things. You might make a detection of something, and you might do interpretations, but it takes a long time to really understand. There's never just, I would say, really never a eureka moment where you point the telescope and you understand it. There's always, sometimes decades, or in the case of comets, for example, centuries before you actually understand what you're looking at. If you want to know more about that, you can read this great book on discovery, classification, and astronomy, where I talk about this idea of extended discovery. The other thing I wanted to say about discovery is that it makes almost no sense to ask the question, what is the impact of discovering life, unless you specify the scenario, right? The impacts are going to be scenario dependent. And here, one of the way I parse this is by doing a little matrix here. Is it going to be terrestrial or extraterrestrial? Is it going to be direct or indirect? And of course, is it going to be microbial or intelligent? And if it's intelligent, is there a message? And if there's a message, do you decipher it? And if you decipher it, what does it say? All these things have different implications, right? You can do the same thing here for the discovery of intelligent life, what I call encounter types 1, 2, 3, and 4. 3 there is the one that was done here that Frank initiated with the radio attempts at radio detections. So all of these have different impacts. You can also look at this in terms of science fiction, because science fiction has looked at all of these kinds of scenarios, and I think the best of science fiction gives us some ideas about what the impact might be. I don't have time to go through this. You could spend a whole talk or days talking about what these potential impacts might be. But the point is that the impact is scenario dependent, right? Okay, so you have to consider the scenarios. Analogy is the third one: history, discovery, analogy. How can we use analogy? A lot of people think analogies are wishy-washy kinds of things. People like Douglas Hofstadter, who's written a 600-page book about analogy, and this is also a hot topic in philosophy of science, think that analogy is anything but a little bitty blip. It's the blue sky that fills the whole sky of cognition. Analogy is everything. We don't even realize when we're using it, but we do. On the other hand, as Catherine says, the problem with analogies is that they're highly persuasive, inherently limited, and easily overextended, right? So we have to be very careful. Can't predict the future. We're not in the business of predicting the future. We're laying out scenarios and guidelines. Astrobiology, after all, makes great use of analogs. All of these analogs that astrobiology science uses. It's just everywhere. So these are just a few of those. You can talk about what kind of microbes would we use then? What kind of analogies would we use? There's a microbe analogy. If you discover microbes, I think the discovery of microbes on Earth might be an analogy. And I don't have time to go through that, but it was a long and drawn-out process. He didn't realize at the time how important microbes were as pathogens and all that sort of thing. It took a long time. That might be one analogy. The analogy you always hear about when you talk about culture contacts when you talk about finding intelligent life, are culture contacts. There are dozens and dozens of them just in the Americas. And what you usually hear about are the Aztecs and Cortez, which of course was disastrous, right? But that's just one analogy. There are many, literally thousands of other analogies, some of them a lot more benign, such as the voyages of Zheng He in the 15th century, which were, followed up by trade and various other things, and there are even monuments now to to Zheng He and some of the places that he visited. Very different from the analogy that we usually hear about Cortez and the Aztecs and things like Stephen Hawking used to bring up, they're coming to get us. That's a very simplistic kind of conclusion. So they're not necessarily destructive culture contacts. You can learn things about language and communication problems. Early actions are important, the need to prepare European conceptions of the other, and you can look for common elements of contact, but not the details. If you actually get a message, here's an analogy, the transmission of Greek knowledge to the Latin West by way of the Arabs in the 11th and 12th centuries. That's a well-known and highly studied phenomenon, but some people, in that case, we knew what the languages were. Some people think that the decipherment of Mayan glyphs would be better, and if there's information flow, even Gutenberg and the printing press, or the internet. And you can see how difficult it is when you talk about what's the societal impact of the internet now, when you're in the middle of it. It's a very difficult thing to parse. One of my favorite ones is the worldview analogy. If you consider the discovery of life as a worldview, perhaps it will change our worldview. And here, we can talk about the Copernican, the Darwinian worldviews, and other worldviews, which have been studied in all their various phases, and that's I think that's one of my personal favorite analogies. The Goldilocks principle, just basically be very careful how you use analogies. Always point out how cautiously you have to use them. And in the book, I talk about a hierarchy of worldviews ranging from the cosmological to the philosophical, theological, ethical, and the cultural. And we're going to hear about some of these over the next few days from various people here in the room. Okay, I'm getting close to the end of my time here. I think I'll skip over this one. History, discovery, and analogy. These are some of the conclusions. The lessons are difficult to learn. They may be ambiguous, but history, discovery, and analogy offer a grounding and experience. We can't make predictions, scenarios, and guidelines. Analogies can be powerful, as powerful in history as it is in science, and the discovery might well be unique, the discovery of life, but the alternative is to do nothing, right? So I think it pays us to think about these things ahead of time. Even if we never find life, these are good thought, experience, and that's why we need scholars in the humanities, social sciences, and philosophy. Let me just conclude by saying, I used to show this graph all the time, astrobiology as a discipline, with the convergence of planetary science, planetary systems, origins of life, and SETI. What I now would like to emphasize is that we also need to add the social sciences and the humanities, which can shed light on underlying assumptions, strategies, ethics, message construction, and societal impact. And also the media. We've seen how important the media is, so we really need to educate the media and bring the media in to help to understand this problem. So, just to reiterate, need to think deeply about our underlying assumptions, need to put philosophy, humanities, and social sciences to work for SETI and astrobiology in general, both for strategy and for impact. Need to lay the foundations now for new disciplines, astrocognition, astrotheology, astroethics, some of these which Ted has pioneered, and we'll hear about more. And the idea of consilience. There's no discipline that is more able to bring together all the disciplines, not just the natural sciences, but also the social sciences, than astrobiology. Earthshots and Moonshots. I would just say that continuing to integrate the humanities and social sciences into astrobiology, not as a peripheral activity, but as an essential one, would be a very valid Earthshot. A Moonshot Well, maybe would form a center for the study of cosmos and culture. Not only the impact of astrobiology, but the impact of the cosmos on our culture. As you know, remember Carl Sagan already talked about the cosmic connection, and I think those connections are growing all the time, and certainly will grow more if we have success in astrobiology. And all of this would be in the spirit of Barry Blumberg, who was very much interested in these impacts, and his, his chair at the Library of Congress is carrying those things on. I think I'll stop there. Thank you.
We have time for one quick question while Rick comes up and sets up. And I saw David's hand go up first.
Yeah. Steve, that was a really interesting talk, and I'm wondering, what really is the lesson of the Mars rock incident? I mean, there's this trope that we always hear, and some of us have written about, that even the discovery of microbial life elsewhere that's convincing will catalyze a cultural change and make us realize our unity and all that stuff. And
Right.
a lot of us want that to be true because we want something to catalyze that change. And yet, and as you described, that's probably the best recent analogy of something like that happening. And although you showed that impressive picture of all those photographers at NASA headquarters,
Right.
and certainly it was a big headline, but then the public kind of forgot about it, I would say.
Right.
And I would say that even if the scientific consensus hadn't switched, which it did, and I'm not part of that consensus, I'm still an agnostic, but that's another question.
Right.
But that maybe the lesson is that catalyzing of a sea change didn't really happen, and that people think it's cool, but beyond thinking it's cool, it doesn't really have that much of a societal effect.
Well, I think that's true with the Mars rock. And I think you also have to parse the short-term consequences with the long-term. And there are all kinds of other things that you would have to consider. But again, this is just, we're not predicting what would happen, but I think to talk about the analogies and the disanalogies is well worth it in terms of future guidelines rather than having to think about it when it happens. Yeah. So as I said, the implications are ambiguous. Yeah.
If it's super short.
I heard a quote the other day which I think applies to the analogies, and that is with models. And the quote was, "All models are wrong, but some of them are useful." And I think that applies to the analogies.
Yeah. I agree.
Great, let's take them again. And next we have Rick Carlson from Carnegie DTM, who will be talking about the origin of the moon.
Yeah, I'd like to thank the conveners for inviting me to participate in this. It's not my general field, but the discussion so far has been pretty interesting. The rest of the program looks really good. You might be wondering why one wants to talk about the origin of the moon at an astrobiology conference, given that you already know from the ballots that we presented that the moon has no life on it, right? You were not given that as an option, although you can write it in. So the fact that the moon is lifeless is probably something the astronauts would have liked to know before they were put into quarantine when they came back from the moon. But it is, we're 7 or 8 hours away from the 50th anniversary of Neil Armstrong stepping foot on the moon. And I thought it was a good thing to sort of explore what we learned about the processes of planet formation from this first adventure of human field geology on another planet. So I'll be looking at the origin of the moon in the context of the 4 models that are probably the most prominent models for the origin of the moon: fission, capture, giant impact, and co-accretion. We'll look first at some of the history of how these models came to be, because they give some good ideas about the thinking and the data that went into the formation of these ideas about how our neighbor was formed here. So the next 3 slides are sort of a summary of a couple of papers from Stephen Brush. These are very interesting if you're interested in selenology and the thinking about moon formation. I think the first interesting aspect is the discovery by Galileo of the Galilean moons of Jupiter. This was at a time when people were thinking about, we just transited from everything orbiting the Earth to everything orbiting the Sun, and then you see these round objects that are orbiting Jupiter. So it gave you the idea that the Sun is not the only center of the universe, is that something like the moon could in fact be orbiting the Earth. So this was at a time then, as I mentioned, when the people are reconsidering the way that the solar system is formed and the structure of it. So there are people, Descartes and Laplace, for example, are thinking about what we now know as planetary nebulas, circumstellar disks, planetary vortex, I like that term. And that basically you form the planets in this disk that's surrounding the sun, and you could do exactly the same thing around a planet. You had a planetary disk and you form a moon in that kind of a setting. What started to move away from that then was actually observations of the moon. Very careful observations were showing that the speed of the orbit was increasing with time, which means the moon was moving away from the Earth. The explanations started looking at both solar and terrestrial tides, and it became fairly physical when people noticed that you can lose energy by tidal dissipation within a solid object like the Earth, or fluid object like the Earth, depending on what you think of fluid. So the tidal dissipation slows the Earth's rotation. In order to conserve angular momentum, the moon has to move out. This also then the tidal locking causes synchronous rotation of the moon, so that we only see one side of the moon as it orbits the Earth. But this then, it doesn't take much thinking to say, oh, if the moon is moving away now, if you go back in time, it was probably moving towards the Earth, right? So George Darwin, the son of Charles, was the first one to suggest that if you extrapolate that back in time, there was a period when the moon was in fact part of the Earth. So this is the origin of the fission model, where the Earth was spun up by solar tides and eventually just basically threw off a part of it to form the moon in one fell swoop, as that cartoon at the beginning suggested. It wasn't really Darwin, but Oswald Fisher who suggested that this ejection of the moon left big holes in the Earth, one of which was the Pacific Ocean basin, which actually was an idea that couldn't be disproved really until the 1950s and 60s, when the theory of plate tectonics came along, and we had some good geochronology where you know that you can't get a 4.5 billion-year-old moon out of a 180 million-year-old ocean basin. So that was the end of that idea for it. But this idea also then fell prey to physics again. Edward Rush and noted in particular that if the moon left the Earth, it was at one time within the tidal radius of the Earth, so that it would have been disrupted. Tidal forces would have been stronger than the strength of the moon, and it would have broken up into a number of pieces, and hence ended up looking something like a circumstellar disk again. So we're moving away from basic fission back into the idea of co-accretion, if you will, or evolution in a circumplanetary disk. But then some more observations of outer planets. Thomas C. noted that there were retrograde moons running around Jupiter and Saturn, and if they're retrograde, it means they were probably captured. So it brought up the idea then that the moon was, an extra object that had nothing to do with Earth formation, and in fact was just captured by the Earth's gravity. Harold Jeffreys, a famous geophysicist, thought initially that fission was a good idea, but then again, this issue about how effectively planets can dissipate tidal energy brought back the idea that solar tides could not spin up the Earth and cause it to distort sufficiently to actually spin off a planet. So he left the idea of fission and started to think more about capture. It was really the father of modern planetary science, Harold Urey, who sort of put the pin in it and said, well, the moon's probably a captured primordial object. And how he came about this, it's an interesting story that I actually wasn't able to figure out from reading these papers. So Urey has the moon being a very cold object that accreted very slowly. This is partly the models of the time for how planets came together, so that the only energies involved in kinetic energy were small asteroidal impacts into these objects as they grew. So basically there was not enough energy deposited in a short enough time to dramatically raise the temperature of the planet. So you see these are quotes from Urey's papers in the 50s. Heat balance in the moon indicates in the interior was formed at temperatures less than 300 degrees C. Urey thought that the Mare basins were in fact at one time oceans. They were not now. He didn't think there was water there, but he thought that the flatness and particularly the darkness of them was related to sediments depositing in an ocean. The other aspect that led Urey to this is that the albedo of the moon is surprisingly low, so it's dark, if you will. And the darkness of it, Urey, of course, was a big person studying meteorites, and the dark types of meteorites are carbonaceous chondrites, which are the most primitive of the meteorites that we have. So these are the kind of ideas that were leading into Urey's idea that the moon was a captured object. It was probably an outer solar system object that just formed at low temperature and got captured by Earth's gravity. So So that's the history of where we got to this. Urey actually was one of the early proponents of going to the moon because this would be a great place to go and collect the primordial material so we could figure out what the Earth was made of. We'll see that actually didn't work out very well. So let's start looking then at some of the data, what we do know about the moon. So here's I think a very pretty slide about the fact that the moon is unusually large. So moons are all in scale relative to the Earth at that scale. And you can see the moon is approaching the size of the Galilean moons of Jupiter, not quite that big, but Jupiter, of course, is huge. So the moon relative to the Earth is quite a large object. The only thing that's close to that actually is the Pluto-Charon system, but since Pluto's no longer a planet, we don't have to worry about that in this discussion. The other thing that we knew about the moon before the Apollo program was that it was a very low density object. So it has a density, uncompressed density in the 3 grams per cubic centimeter. And what this does in an airless body, at least, is it allows you to say that there's probably a deficiency of iron in this body. So here's a plot. This is the atomic ratio of iron to silicon. Doesn't really matter what oxidation state the iron is in. Solar composition is shown by that yellow band across the middle. You see that most of the planets, Venus, Earth, Mars, are more or less close to solar. Most of the meteorites that we have, the primitive objects in the solar system, are also very close to solar in their iron to silicon ratio. Some of them have figured out a way to lose iron. But then you look at both the moon and Mercury. Mercury's got an incredibly high abundance of iron, whereas the moon has a very low abundance of iron. So this sort of throws, in my opinion, a monkey wrench in the idea that this is a primitive object to begin with, although Urey explained this away as using a model that was very popular when I was a student, actually in the 70s, called heterogeneous accretion, where planets actually grew by compositionally distinct objects at different times. So you think about the structure we know now with the core in the middle. You started a planet with an iron core and grew a silicate body around it later. In fact, in Urey's idea, it was the other way around, where silicates arrived first and then you put iron on top of that, which brought up all sorts of problems because nobody could understand the physics of how iron deposited in the surface of the planet could flow through a solid silicate planet to get to the core. So there were lots of debates about this argument, which don't need to be held because that's not the way planets form. So this brings up the idea of how do you separate iron and silicon? It turns out that the physical properties of these two materials are so different that it's actually pretty easy to do it. The way you do it is by big collisions. So here's an example of some hit-and-run collisions from Eric Alsfag's work. These objects there's 2 different types of collisions shown, the upper and the lower band. This shows 2 planetary objects with blue represents the rocky part and red are the metallic cores. So on the top, these guys run into each other. This is truly hit and run. It's kind of a glancing blow, tears both planets apart, throws a lot of silicate out into space, most of which ends up back on the planet, but these then go on their way and they're modified a bit in their iron-silicon ratio. In the lower one, it's a different kind of impact. A smaller object hits a bigger object. You see it just tears the small object apart. All the red material in the small object will eventually stream back to form, to go into the core of the large object, and you're left with this big debris of silicate material out in space. So you think of that, you've just enriched the impacted object with iron, increased its iron-silicon ratio, and the stuff that's going out into space has very little iron in it, so you've created an object with a low iron-to-silicon ratio. So this is a great way to separate iron and silicon by processes that we know happen in planet formation. But the important aspect of this is that this is a very energetic event. So you can imagine, if a planet looks like this, it's not forming at 300 degrees centigrade, right? So these are very high temperature events, and that's where the Apollo program came in. So the expectation from Urey was you're going to land on this, you're going to be picking up primitive carbonaceous chondrite material, you're going to find pre-solar grains, you're going to find all sorts of fun stuff on the moon. But what we found instead was a very high temperature object. So even just from a handful of grains, in these first papers from John Wood and colleagues, they actually just picked up it was a soil sample, and they noticed that there were chunks of this rock type called a northosite, this white guy at the end. The northosites are not unknown on the Earth, they're not particularly common, they're not that rare either. They're composed of basically a single mineral, anorthite feldspar. And it's not that easy to get a melt of planetary composition to form a single a rock made out of a single mineral like this. But one way that you can do it, by analogy with various examples on the Earth, is you do it by melting. So if you have a melt of rock of a composition expected for the bulk moon, and you allow it to crystallize, the first thing that crystallized are magnesium-rich minerals like olivine and pyroxene. They're denser than the liquid, they sink. that eventually enriches the magma in elements like calcium and aluminum, which are the ingredients of feldspar, and that anorthite feldspar then actually, when it crystallizes, is less dense than the liquid, so it floats to the top. So what you end up here is, I like this cartoon, it's from John Longing from a long time ago, but you have this idea that basically you're floating crystals up to the top, and you're forming things that they're called rockbergs at this stage. Those will eventually coalesce to form a very thick anorthite crust around the whole moon with this magnesium-rich silicates in the middle. But what this is telling you, obviously, is that the whole moon had to be melted. So if the whole Highlands crust of the moon is anorthosite, the whole moon, or a very large proportion of the moon, was melted. So the very energetic birth to the moon. And it turns out that, from a handful of grains, these guys extrapolated not only that all the Highlands, Apollo 11 didn't land on the Highlands, right? So there's sampling stuff that didn't even come from the landing site, really. Came to the conclusion all the Highlands are anorthosite, and it covers the whole moon. Not only that, but if you think about the density difference between anorthite and the basalt lavas that the Apollo 11 landed on, they did the simple buoyancy calculation, said, well, that means that the anorthitic crust is about 25 kilometers thick. We now know that's about 30 kilometers thick from newer gravity measurements. So this, this to me is a really stunning, huge extrapolation, but they were right. So here's some good examples of global mapping from orbit in the moon. So the top one is iron concentration. The lava flows where Apollo 11 and most of the other Apollo and Luna missions landed are iron rich, so they show up in red on this figure. This is the near side of the moon. But when you go to the far side of the moon, it's mostly iron deficient, shown in the blue color there. The other interesting one that will be important for a couple of slides, this is thorium. So thorium is interesting because you can measure it well from space because it's radioactive. You can measure the gamma rays from an area like this. What you see is on the moon, the thorium abundances are much, much higher only in this small area of the Earth-facing side of the near side of the moon. And we'll get the importance of that in a bit. So this totally transformed idea of the moon from being this primitive cold object into being something that formed extremely hot above the melting point of silicates. So there are other aspects of this that are very important in terms of what the planet ended up with. So here's an example. This is a plot of potassium versus thorium. The reason for doing this is both of these are radioactive. They can both be measured from space in airless bodies anyway. But the difference between them is that potassium is a relatively volatile element, has a half condensation temperature of 700 degrees centigrade, whereas thorium is a very refractory element. Condensation temperature is something like 1400 degrees. So the important part about here is the ratio of potassium to thorium, which is shown by the various lines. So you have the data from Mars now up there. We have the data from Earth. There's of course thousands of points on that, but I just drew the line. We now have data from Mercury. This is actually from the Messenger mission work to put together this plot first. So both Earth, Mars, and Mercury have more or less the same potassium-thorium ratio, at least within our degree levels. And the Earth potassium-thorium ratio is about 3,000. It gets up to about 5,000 on Mars. But the interesting aspect of that is the solar composition. The sun has a potassium-thorium ratio of 18,000. Okay? So you basically you've lowered that ratio by a factor of 6, which means you didn't condense much of the potassium in the Earth. So if you think of potassium as a volatile element, what does that mean about water abundances in the Earth, carbon abundances in the Earth? So these are clear signs that the formation of the terrestrial planets anyway occurred at very high temperatures where you're not condensing most of these volatile elements. The moon, it's even worse. So the moon has an extremely low potassium-thorium ratio, which again is probably testimony to a very high temperature origin for it. So we think about how do you make a planet hot enough to melt. There's two ways that you can do it. One is you can do it with radioactivity, but in order to do that, you have to have a radioactive isotope that has enough energy over short enough time to raise the temperature of the planet above melting. We actually know that happened because we have data from various iron meteorites, as shown in the white plot up there. Some beautiful work from Thomas Kruger, where looking at iron meteorites, we know that the iron from silicate separation, which probably represents core formation on these bodies, probably asteroidal-sized bodies, occurred within about 500,000 to 2 or 3 million years of the beginning of solar system formation. TCAI is calcium aluminum rich inclusions. These are the oldest objects in the solar system. So you see here that you're forming, you're melting these planetesimals within about 500,000, a few million years. And in order to do that, you had to grow the object big enough by a few 100,000 years in order to get it hot enough to retain enough heat to melt. And it turns out that that kind of time scale is a perfect time scale for heating by aluminum-26, which is a radioactive isotope. It's a major element. It has a half-life of about 750,000 years. So if you can grow planets on that time scale, great. You can melt them, no problem whatsoever. So a lot of the planetesimals in the Earth that form the terrestrial planets probably had a melting episode to begin with, driven by aluminum-26 heating. The other way that you can do this, of course, though, is the kinetic energy of impacts. There's no real time constraint. That's just whenever that impact is going to occur. So it turns out that we can date that on the moon. And we can date that because when you have this magma ocean and the crystallization of it, this, of course, is a classic wet chemistry technique, recrystallization to purify the element that you're after. Well, the moon did it for us. So it has this mantle that's composed of magnesium silicates. That's that green pattern for these kind of elements. These are elements that don't fit into those minerals very well. The white is the plagioclase-rich crust, the feldspar-rich crust. Doesn't fit into that. And then it's left over with this band of residual liquid in the middle that's called creek because it's enriched in potassium, rare earth elements, and phosphorus. So these have these very different patterns of these elements. And what radioactive systems date is actually chemical changes like this. And it turns out that most of these elements are a large fraction, uranium, thorium, potassium, and the sister of potassium, rubidium, are all radioactive. Samarium and neodymium are both radioactive. So, or samarium is anyway. So you can use these elements in particular to determine the ages of this event. So one of the beauties of the magma ocean model is that it says that all these reservoirs on the moon form more or less at the same time. So the crystallization of a magma ocean like this occurs in hundreds of thousands of years, at most a couple million years. So the kind of precisions that we can get with geochronology, that's more or less instantaneous. So we look at the ages of lunar crustal rocks. There's some range in them here, but there's some debate. I mean, the lunar surface has been beat up a lot by meteorite impacts. So some of this is simply just disturbance and inability to get accurate ages. We've been trying to refine those ages. The red points are done with more recent approaches to this. You see that they're coming down to a rather tight range at about 4,300 million years, 4,400 million years. That gray band is actually a different approach to it. It's measuring ages for this creep component, which can be done more convincingly. But you see that's also about 4.4 billion years. And the most convincing comes from data like this. These are, the 142 neodymium is created by the decay of 146 samarium. That's a 100 million year half-life, so it was present when the moon formed, but it decayed away within a couple hundred million years, a few half-lives. The fact that you have this variation, 142 neodymium relative to stable isotope, and it correlates with samarium-neodymium ratio, says that this change in samarium-neodymium ratio in all these lunar rocks occurred while 146 samarium was still alive. So you see they all more or less fall along a single line, and this plot includes not only mineral separates from the crust, so these are the FAS is ferro and anorthosite crust, so these are rocks that have been dated, but also two types of Mari basalts, low titanium, high titanium Mari basalts, and creep as well. So all these rocks fall on the same line that gives an age of about 4.3, 4.4 billion years. So the key to that then is that that's not 4.56 billion years. That's way too young to have aluminum-26 as a heating mechanism to melt the moon. And then you go to the Earth. So a giant impact into the Earth, if that's where the moon came from, should have left an impact in the Earth. And you can sort of see that here. To determine the age of the planet isn't that simple. We're starting to find rocks that are older than 3.8 billion years. There's 4.4 billion year old zircons in Australia. There's a 4.3 billion year old group of rocks in Hudson's Bay. But if you look at that plot there, that's a bunch of different ways that have been used to determine both the age of the moon and the age of the Earth. And the thing I'd like you to take away is that little gray band over there is the age of the solar system, and none of those ages approach that. So the Earth and the Moon system appear to be quite young. More than that, we have data now that shows that the Moon is made up of material that most likely originated from the Earth, and we have these various isotopic tracers that allow that. This one here is oxygen, titanium, chromium, tungsten. I don't have time really to go through why they're this way, but the blue points are the Moon, the black line is the Earth, and you see the blue points for the Moon come very close to the Earth composition, whereas meteorites in these gray bands describe a wide range. So if the Moon was an independently created object, it could lie anywhere in those gray bands, but it doesn't. It lies essentially the same place as the Earth. So that brings in these kind of models, giant impact models from our postdoc Miki Nakajima. These are always fun to watch. So these are incredibly violent impacts. And I'm showing you this one because this is a traditional model. So the moon is eventually going to form out of all this stuff that's now forming a disk or ring around the Earth. But the interesting aspect of this is that almost all of that material that's in orbit around the Earth came from the impactor. It didn't come from the Earth. So that then is going to violate the information I showed you on the last slide, which drives you to more energetic models like this one from Sarah Stewart's group, where this is a very violent head-on collision that basically mixes the two objects to the point where you can't tell the difference. They're color-coded by core and mantle. Let that go. So an interesting aspect I mentioned early on, so you see that light blue around the core, that's the core of the impactor, goes right to the core of the Earth and stays there. So what's going to end up in orbit around the Earth is a mixture of silicates that comes both from the Earth and from the impactor, and the mixture is so good that you probably can't tell the two apart. I'm running out of time, so I want to do the last couple slides that really have something to do about astrobiology, and that's the rate of impact. So impact is clearly a very important process in the growth of planets. We know from the surface of the moon about the impact record. We think that it's some sort of a decline like this through time. It's the number of impacts per surface area per time interval. And what was found on the moon is this thing called the lunar cataclysm, or the terminal lunar bombardment, if you will. And there was a time when this was a big thing in the early 70s and 80s. It came out the oldest rocks on the Earth at 3.8. We now know there's older rocks, and that's the point for the earliest life on Earth as well. So this came from dating of lunar melt rocks, this is the actual data for it. You can see there's this big peak at 3.87. So one of the explanations for that peak is that it was a time when the moon's surface was highly bombarded probably to the point where it was a sterilizing event for any life that was trying to form either on the Earth or the moon. So that's one explanation for it. The other explanation for it is that, in fact, this represents a time when you've got a very steeply declining rate of impacts. This is a graph that shows you how much of argon is lost. Potassium radioactive that decays to argon. That's one of the dating tools. If you've lost the argon, you're getting too young in age. So one of the explanations for this is that the 3.8 billion years is just the point where you basically stopped erasing all the older evidence. And the other explanation for this, which is a good reason to go back to the moon, is that what you're dating here at 3.87 is in fact a single impact. It's the age of the Imbrium Basin, which if you look at impacts of that magnitude, scatter material all over the surface of the moon. So given that the lunar program, Apollo program and Luna programs only land in a very small area of the moon, it's quite possible that that peak simply represents a single event in moon formation. So I had to zip through that, but I mentioned at the beginning that of those 4 models, it turns out that 3 of them are probably right in some sense. Capture doesn't seem to fit much of anything, but in models like this, what you're looking at is the moon forming from a vortex, if you will, a circumstellar or circumplanetary disk that's caused by an impact that fissions material off the Earth. So in some sense, all 3 of them seem to work. So thank you for listening.
One quick question, maybe? Question. Oh, there you go.
The great band you showed with the isotopes showing how different or how varied the meteorites are. If you just restricted that to meteorites which were closer to the Earth, such as ensotites, how narrow would those bands be?
So the ensotite chondrites are the one group of meteorites that fall on top of the Earth composition pretty routinely in all of these.
And the ones whose distribution is closer.
Except for this one. So these 2 are due to nucleosynthetic issues. Ensotite chondrites are close. This is probably due to some photochemistry in the nebula. Ensotite chondrites are close. And this one, this is a radioactive product that's produced by 183 half-king decay. It's a very sensitive tracer of core formation. So what this is tracking basically is the history of core formation on the object. And that would be a very difficult thing to do if the moon were an independent object, and it would have to have the same core formation history as the Earth. And that's really starting to stretch it. These I agree. I mean, this could be like an inner solar system composition, and the impactor into the Earth could have just had more or less the same composition. This one is more difficult to explain that way.
All right, thank you very much.
Yeah, just a quick announcement. We're going to have a few of the flash talks today, starting with Carl. So if you just prepare, I'll read the order of them. Carl Pilcher, Barry Welsh, Jeff Kuhn, Charlie Lineweaver, and maybe Daniela if we have time. Okay? And remember, the rule is we're going to show a slide and you have 1 minute to describe it. But before we start, a couple of announcements. First of all, outside there are labels, blank white labels you can put on your coffee mugs to distinguish one from another. They all look the same. Anyway, take one. The other thing is we will go from here over to the Drake Lounge, which is in the residence hall right above where you picked up your badges. And then there'll be drinks up there, and dinner will start at 6. And you don't have to take your wine glasses down. There are real wine glasses down at the dinner. And at around 6:45, we should all move over to the Science Center, which is a big building there, because there'll be Chris's talk at 7 o'clock. And the last announcement is when we get done with here, before the drinks, everyone who is staying at Mountain Quest, would you please come up here and talk to me? Okay, we have some arrangements.
Gee, what about dinner? Is drinks and dinner the same?
Drinks are first, and then dinner. Well, there will be wine with dinner.
What time is it?
Dinner's at 6.
Drinks?
Drinks are right
Drinks are
after.
5:30 when this session is over, or whenever this session is over and you get over there.
Okay.
Okay, I've got a stopwatch here, I think. I just got this, so here.
It's not going to be a lot.
What's the camera? There we go. Okay, Carl, come on up. You've got a minute starting
Okay,
now.
I want to pose a question to this august group. Does SETI equal SET-C? I'm not proposing I know the answer to this question. SET-C is a search for extraterrestrial consciousness. Another way of posing the question is, are we imputing consciousness to the creators of technosignatures? So it's important to distinguish between intelligence and consciousness. I've used two definitions that I don't think are terribly controversial. There are lots of definitions. So intelligence, the ability to solve novel problems, new problems in a changing environment. Consciousness, the state of having subjective experience. And I think I'd remove awareness because that's a subset of that. So if SETI equals SET-C, and if it doesn't equal SET-C, what are the implications? And I've just put up one possible implication. For example, going back to Lynn's talk now, are we assuming convergent evolution of ETI consciousness under, for example, the influence of selection for traits that may have led to consciousness on Earth? And a couple of those that have been suggested are error correction and modeling of attention.
Thank you. Barry Welch, yes.
Hi.
And begin.
Right, about 40, 50 years ago, Towns and Schwartz said that perhaps advanced civilizations might communicate via lasers. So we have nothing better to do in my retirement. I thought of a really great way to do this. We happen to have an image intensifier on a 10-meter telescope in South Africa, and we thought we'd have a little look to see if we can find pulsars coming from some objects. Which objects would we look at? We'd look at stars that have no planets in the habitable zone. The thing is, you've got to get rid of the huge amount of light from the star to see the pulses. However, because we use photon counters, which we can bin the data to picoseconds, the laser pulses should be brighter in that instance than the stellar photons. And we can discriminate through a thing called pulse height distributions, 10. Okay, there's a couple of papers out there, and of course, I'd be in Stockholm now if we'd seen it. Thank you.
So I thought that I was going to say something clever about the anthropomorphic arguments, but I unexpectedly got off a plane from China just 3 hours ago. Therefore, just read the last section, the last bullet here. The point of my being here is to try to make the argument strongly that these questions about anthropomorphism are actually experimental questions, or could be within 5 to 10 years, and that's what I'm going to talk about on Tuesday in more detail.
Hey, thank you. Charlie.
All right. I thought, since we're talking about life, that we should see what type of diversity there is in life. This is a new phylogenetic tree. Lynn showed one from Pace, so it was from 1997. This is one from Castell and Banfield's group, 2018. And one of the most, for me, a remarkable discovery has been the CPR. These are candidate phylum radiation up here. and here in DPAN. So there are two things. One is that, I should also mention that the eukaryotes seem to be rooted inside of the archaea, so we, in some sense, you can consider yourself to be an archaea. And also, these things are also deeply rooted and monophyletic. Now, I should point out that this is so important because the closer and deeper we get, into this tree, I think the closer we get to chemistry and physics, which I think we could all admit or agree that that is universal. But biology, whether it is or not, we're not quite sure.
And Danielle.
Yes. Yeah. So I thought I would just introduce a little bit my talk on Monday by trying to look into ways we communicate and looking at other species that communicate in a very different way. Like the snoutfish, for example, the species uses electrical signals, and if they were communicating via radio, they would have a much simpler way to communicate. They wouldn't have to convert their signal first into an electrical one. So by looking at how other species communicate, we can also understand perhaps better how other intelligent life forms on other planets might be communicating. What is interesting about the research related to this fish is that also scientists detected a way how they can filter out electrical noise that is not interesting for their survival. And that led also to better understand how we filter out noise from our body and for survival.
Thank you. Herschel, you are next.
I'm next?
Yes, if I can understand what I'm going to do.
It should really have been 90 seconds.
All right. So, start. Okay. So we're looking at simple molecules in diffuse interstellar clouds, and the molecules we're studying are very simple hydrocarbons, with three carbon atoms and a few hydrogen atoms. And my recent work with the GBT has been to study these simple hydrocarbons in absorption, and the absorption spectra are shown on the right. The question we're trying to answer is whether these simple hydrocarbons are produced exclusively from simpler ingredients or whether they break down from larger molecules. Now, the diffuse gas is a very rich laboratory where things as large as the Buckminster fullerene ion, C60+, has been found. So the question we're trying to answer by looking at diffuse clouds within the galaxy is whether the chemistry happens from the bottom up or whether it happens from the top down. And we're planning to do a survey of these bright H2 regions, using them as lamps to study molecules and absorption and answer this question.
Thank you.
And if you'd like to know more,
Kelly Smith. And on deck is John Chambers.
Okay, so I'm Kelly Smith, and I'm the president of Social, it actually has another S on it, the Society for Social and Conceptual Issues in Astrobiology. It's a new organization started in 2016. It's got about 150 highly interdisciplinary people, and it's basically for anyone who's interested in seriously thinking through some of these broader extra scientific questions involving astrobiology and space exploration. So it's open to anybody in here, whatever your disciplinary background is. Our next conference is in March at the University of Mississippi, and the call for papers is out. The simplest thing to do at this point is just email me. Here's my email, and I will send you the call for papers, and it'll also be posted on NASA's site and stuff soon.
Thank you. And by the way, all of these will be on the website. Okay. John Chambers.
So one possible explanation for the lack of positive results from SETI is that life has to go through a series of bottlenecks or hard steps which have a very low probability of occurring. Here's a Monte Carlo simulation of billions of stars with 5 hard steps. And you can see the timing of the hard steps in a successful case is roughly evenly distributed through time. That naturally explains why life occurred very early on in the history of why life formed early in the history of the Earth, but intelligent life formed pretty much at the end of Earth's habitable lifespan. I've also looked at the effect of different stellar masses on this process, concentrating on stars between half and one stellar mass, noticing that different stars have different lifetimes and different frequencies. And you see there's a very strong bias towards very low mass stars. Now, given that we formed on a solar mass star, which never occurs at all in this survey, that suggests there's a very strong reason why life cannot form around, intelligent life cannot form around stars less than one solar mass. Thank you.
James Schwartz and Catherine, you're on deck. James Schwartz, yeah.
Oh, well, that's the first page for. Oh, that's the first page for my talk on Monday. But I do have something I can say. So I'm helping to run an interstellar workshop in Wichita in November. I've got cards out. So if you're interested in interstellar stuff, please submit an abstract. I'm also doing a NASA Celebration Conference in October. Talk to me if you've got an idea for sort of a talk celebrating NASA science or Apollo.
Thank you. Catherine.
That's not me. Okay. Oh, that's me.
There you go. Oh, who's there?
So this is an intro for my talk on Monday.
This is your. This is your.
Yeah.
No.
No, this one.
Next one.
Okay.
Yeah.
This one.
Okay.
Yeah. So I wrote it down so that I'll be very fast. So usually in this space we talk about communication in the context of SETI and techno-signatures, but I want to draw your attention to the fact that we can use communication as a liaison between biosignatures and techno-signatures, because communication is a complex system that exhibits the co-evolving and emergent patterns of the communicators, whether these communicators are cells, organisms, or AIs and their environments. So whether we are talking about the physical aspect of the organisms for which communicators are embedded in the physical world, as our perceptions, for example, to communication where, or whether we are talking about social organisms that have evolved cultures and their communication is embedded in their cultural context, or whether we are talking about cultures that have evolved technologies and their communication is embedded in the full biological, social, technological ecosystem. And their communication is highly correlated with the collective behavior of life from multicellularity to society.
What's the next one?
That's me.
Oh, okay.
It's always a hard act to follow. Okay, so a work in progress, alien rights observers, is just something I'm throwing out there just to get the conversation started. So astronomers, you study things that are very, very far away that you don't have any hope of getting to. However, as soon as you find a planet that is bearing life or something like that, other people are going to be working on propulsion systems to get us there. And I'm not comfortable believing that 100 years from now, we're not going to be able to reach any of these things. I'm also not comfortable just extrapolating our rules for microbes, which are basically, oh, they could kill us, okay, we'll kill them. We have to think about the middle range. So we got to think about the rights of life. How do we incorporate that? So when the NASA AstroBioology Institute was constituted, there was conscious thought about how do we work together, how do we articulate our disciplines. In this particular case, I think we need a mechanism, because otherwise meetings like this may not necessarily include somebody who's thinking about the interests of any alien life we might encounter in the deep future. So I thought, human rights observers, alien rights observers, I don't know, it was late, but let's work on it. Let's think about how we're going to do this.
Okay, yes. Andrew Simeon asked me to talk about this very briefly.
Oh, excellent.
So the Breakthrough Listen Project is an international effort using a lot of really big telescopes at multi-wavelengths in the radio, optical, and even soon, I think gamma rays, well, using gamma ray telescopes at least. And it uses commodity off-the-shelf technology and a Silicon Valley ethic in order to do this. And it's a very scientific way about going for the search for extraterrestrial intelligence. And it's privately funded. And it's also using the Green Meg telescope right here in the backyard. And Andrew will be here to talk about it. And there's others in the room, like Dan, who are part of the project, and you can talk to them about it as well. Thanks.
Thank you. Martine? Martine? Oh, you do? I can't believe we're keeping this on schedule. This is really
5 fundamental aspects of reality. First, I think most of the universe and most of all reality is just waste. There's more atoms in nebula than ever needed for the stars that we end up having to produce planets and life. Secondly, some kind of code. Everything is tied together with physics. Physics is code. I had a great presentation earlier today how life kind of spontaneously generates at least an RNA code, carbon-based life. After that, diversity. Everything that exists starts mutating and changing. That leads to stupidity. Einstein said that he was only sure about 2 things. The universe was infinite, human stupidity was infinite, he wasn't sure about the universe. And finally, grace. Notwithstanding all of the stupidity, there's enormous acts of kindness and beauty, and this conference is, to me, an example of grace.
Thank you. Me. There's no need for time. Yes. Oh, this is quick. Because the point is that if you're out there and you want to attract another civilization, you want to be attractive to astronomers because we're the only ones that are actually looking and paying attention. So how do you attract astronomers? Well, you create something that they would find in the course of their research, and then you make it interesting to them. For example, you create a pulsar-like signal. We search for pulsars here all the time. Astronomers will detect the pulsar, and then, for example, if you decrease the dispersion measure regularly, that's going to attract the attention of astronomers. It's completely unphysical, and by the time you take that dispersion measure to 0 or negative, you will have gotten their attention. At that point, you can slip in a message. The beautiful thing about this is it has strong implications for our behavior now. We are already searching for pulsars. We should support people that search for pulsars, and we should get more of that going. But as important, we need to archive that data. This data has been flushed in the past because we couldn't afford to save it. And as new tools of analysis come along, it's possible to do the search on this unique data set. So I'm saying support it, find a way to keep the data available for future researchers. Thank you.